The following podcast is intended for informational and educational purposes only. Please speak to health professionals before making any medical decisions.
Gene therapy research can feel like a complex topic, and one that yields many questions. Why are scientists researching gene therapies? How are they designed to work? What are the unknowns? Let us answer some of your questions here, and it's in the Genes podcast. We're excited to feature experts in the field, from research scientists and healthcare providers to an actual gene therapy trial participant. It's in the Genes is produced by Bloodstream Media and exclusively sponsored by BioMarin Pharmaceutical. We're here to turn complexity into clarity. Be sure to subscribe to It's in the Genes from bloodstreammedia.com. If you're hungry for even more on gene therapy research, visit hemedifferently.com and sign up to stay in the know on the latest in gene therapy research. Today, we are on the precipice of research into gene therapies becoming an everyday reality for treatment for rare and chronic diseases. As this science becomes part of mainstream conversation, we find ourselves asking, how did this come to be? How did the discovery of DNA and chromosomes and the human genome come together to form a whole new approach to investigating medicine? This is what we will set out to discover over the next 5 episodes of It's in the Genes, a gene therapy podcast. Our journey begins as it did for scientists centuries ago with the complex question, what makes us uniquely human? It's true that some of us have blue eyes, some of us have brown eyes. We have different personality traits and blood types and hair color. As researchers came to discover, the answer to what makes us human is DNA, but this is tricky. We share 98% of our DNA with chimps, for example, so it's not DNA itself, but the differences in DNA. Your DNA is the most unique and identifying factor about you. It helps determine what color your eyes are, how tall you are, and why your feet just happen to resemble your mother's. DNA is the human instruction manual, and genes are a section of DNA which is involved in carrying information for a particular trait, like your hair color or how tall you are. Genes also play a critical role beyond determining physical traits. They help tell your body how to create important proteins. So gene therapy is under investigation to see if we could correct or fix those genes that create important proteins. You see, DNA is constantly subject to mutations and accidental change in its code or sequence. Mutations can lead to missing or malformed genes, and that can lead to disease. Some mutations happen during cell division when DNA gets duplicated. Still, other mutations are caused when DNA gets damaged by environmental factors. Gene mutations can range from pretty serious to not so serious. For example, a mutation of the aptly named FOXC2 gene gave Hollywood icon Elizabeth Taylor 2 rows of eyelashes. Not too bad, huh? But another mutation that has been discovered is located on the dystrophin gene. Why this is so important is our muscles need dystrophin in order to regenerate. So if you have a mutated dystrophin gene, your body lacks this essential muscular protein which is responsible for producing dystrophin. As a result, existing muscle cells deteriorate over time and are gradually replaced by connective and fatty tissue. Muscle strength weakens during the course of the disease. The first symptoms usually appear around the age of 5. Children with this mutation begin to have difficulties with movements they previously completed with ease. For example, climbing stairs or getting up from the floor. At approximately the age of 12, they are no longer able to walk, later losing movement in their arms and their hands. Due to persistent respiratory and cardiac failure, the majority of patients do not reach the age of 40. If you have a mutated dystrophin gene, you have Duchenne muscular dystrophy. The gene mutations we hear about most often are the ones that cause disease. Cystic fibrosis, sickle cell anemia, Tay-Sachs disease, hemophilia, and color blindness, among many others. All of these disorders are caused by the mutation of a single gene. As scientists began understanding more about DNA and how they could manipulate DNA in a laboratory, they began wondering, could we clone certain genes? And if so, could we find a way to transfer them inside a human body? And if so, Can we research them in treating diseases like hemophilia and muscular dystrophy? The concept of gene therapy research was born. Although the technologies necessary to make human gene therapy research a reality are fairly recent, as a concept first introduced in the 1960s, the idea is more than half of a century old. In fact, scientists were able to incorporate functional DNA inside human cells in a lab as early as 1961. However, early attempts at gene transfer were extremely inefficient, and the handful of clinical trials attempted proved unsuccessful. What makes gene therapy so complicated? What have been the incredible successes and the devastating setbacks to this science that is changing the way we research medicine? In order to understand gene therapy, we better start at the beginning. Where did the idea come from, and how are researchers handling the pressures of such complex science? As we enter a new frontier of medicine, there are so many categories to consider. In this series, we hope to give you a glimpse into the world of complex, innovative science and the people making it happen. You'll hear about the clinical trial process and the milestones that have been achieved in the process. You'll hear from physicians and researchers who have been on the front lines of cutting-edge science, and from the patients whose lives have been altered. There's so much to cover. Let's discover the wonder of gene therapy research.
I'm not kidding when I said I had no clue what DNA was when I went there.
In 1991, the LA Times called Dr. Theodore Friedmann Mr. Green Genes of biotech. However, it took quite a storied journey to claim that title. Dr. Friedmann was an apprentice to the famed Dr. Frederick Sanger of Cambridge University in the 1960s, and it was there, in Dr. Sanger's laboratory, that Dr. Friedmann learned how to sequence DNA. Researchers wanted to understand what made up the genetic code on the DNA strand by showing the sequence, or precise order of chemical units. Only then could researchers decipher the genetic function of any gene.
I knew it was going to be the secret for gene therapy, and that we needed to manipulate DNA just like a drug, just like the chemical that it is, but I didn't know what it was. I didn't know it had two ends. I didn't know 5' from 3'. I knew 4 bases, but that's it. I learned about DNA. I worked with Sanger on finishing the sequence of Phi X. My job was to sequence that area and to make sure that we got it right. And I did that, and we did get it right. That's what the year was devoted to. But of course, it gave me a knowledge of how you play with DNA and how you manipulate it and how you change it and how you cut it up and how you recombine it. And that was the technology that one needed to clone the gene. The work that I was doing with Sanger during that second year, it wasn't gene therapy. It was just learning what this tool is, this tool of DNA sequencing.
In the 1970s, while Dr. Friedmann was at UC San Diego, he took what he learned in Sanger's lab and started his own experiments. He began writing one of the first scientific papers on gene therapy. He boldly claimed the idea that the potential to not just treat symptoms of disease with traditional medicines, but to actually fix or override the defective genes with new ones. The concept of gene therapy arose during the 1960s and 1970s. In 1972, Theodore Friedmann and Richard Roblin published a paper in Science called Gene Therapy for Human Genetic Disease, which cited Stanfield Rogers' proposal in 1970 that good DNA could be used to replace defective DNA in people with genetic disorders. Let's hear from Dr. Friedmann himself as he shares his work in the field.
We did an experiment that really is the hub of my, the real origin of my interest in genetic disease, the therapy and gene therapy. Not only to understand these diseases, but maybe even to treat them. That seemed to be an interesting idea. That it would be an extremely interesting experiment to take fibroblast cells from these children and grow them in culture, and then expose them to DNA from normal sources, from parents or from just a normal donor, DNA donor. That would be interesting, and that would be kind of a clue that it might be possible to treat rare human diseases by genetic means, by adding the correct gene. That was not thought of in those days, but we convinced ourselves that there was evidence for the restoration of some production of the gene product of this gene. We could see evidence that some cells seemed to be normalized, but at a very, very low frequency, very poor efficiency. And at that time, we called it transient transformation. It was the kind of an aha moment for me, because it suggested that this confluence of interest in genetic disease and in using genetic technology, proteins and genetic technology.
So while one group of scientists, like Dr. Friedmann in 1972, are concentrating on DNA and cloning specific genes that are known to cause disease, at the same time, another group of researchers are starting to wonder about how to get these newly cloned genes into the body, a delivery system, if you will. It turns out experiments with bacteria started the thought process and ended up with viruses, or a viral vector, as the potential delivery system for these cloned genes.
We were in the era of Star Trek and go where no one's gone before type mentality, and I ended up taking on the project of making a viral vector to treat genetic diseases.
You just heard the voice of Dr. Richard Samulski, and his entire career has been spent developing the viral vector, or what scientists hope would be the delivery system or the transport vehicle for a new functional gene. Let's listen in to Dr. Samulski share his story of being on the forefront of discovering that viruses could be the delivery system for cutting-edge science.
So, a Nobel Prize was just given to a group from Hopkins, and they were, for the first time, showing that you could take DNA from animals and clone it into bacteria, and then the bacteria would amplify and make thousands and thousands of copies, and that would allow you to study it more readily. His name was Nick Muzicka. Nick was trying to develop what they referred to as viral vectors, shuttle systems that would work for delivering genes in human cells the way bacteria plasmids were working in the bacteria world, demonstrating that it could, we could go from bacteria where we would make milligram amounts of the DNA and it was completely inert when you put it in human cells, but if you put it in human cells under the right conditions, it would turn into a virus. And at that point, we knew instantly that we had a shuttle vector that could be retooled to carry genes of interest. And lo and behold, it's 40-something years later, and we're still working on chipping away at some of these genetic diseases.
And the idea was that, let's see if we could learn how to modify a virus to replace its own gene, its own tumor gene, with a potentially therapeutic gene, then create a therapeutic agent out of the virus, and fool the virus into thinking that it was doing some of its own work, and using it as a vector for introducing genes into cells. That field, though, then exploded in the early 80s by people who learned how to take a tumor virus, RNA tumor virus, leukemia virus, and disarm it in a way to remove its pathogenic gene, or to replace the pathogenic gene with a potentially therapeutic gene. And that was the real opening wedge to what we now know as virus vector-based gene transfer for gene therapy.
So, do you have it all figured out? Understand it? Okay, me neither. Let's go back to the basics. What is gene therapy research trying to figure out anyway, Dr. Samulski?
So if you imagine it going into a room and you flip the light switch on and the light doesn't come on, you go, oh, I can't see in here. So the objective of gene therapy is to take that light bulb and replace it. And when you replace it and flip the switch on, then everything functions appropriately. And so what we do is we carry these light bulbs in our viral package, and we go looking for the cells that are not working or lit up appropriately, and we swap them out for the good light bulbs. And when they come on, all of a sudden everything's functional and you can see again, and that cell begins to work appropriately. Now that can be a protein that keeps you from bleeding, like hemophilia, or that could be a protein that allows you to see, like the Leber's congenital amaurosis, or that could be a gene that is used for muscular dystrophy, like the dystrophin gene. And each and every one of those, as you realize when I described them, they are in different locations, either the eye or the muscle or the liver. And therefore, you realize that the molecular FedEx truck needs to travel through the body to get to these locations. So the majority of these deliveries are done by putting it in the bloodstream, which is almost like the interstate system for our body, and letting them get to their locations. Or in the case of like the eye, you have direct access to it, so you can actually approach it from the outside and deposit it in. So you're left with physical techniques to get this to the target, and you're left with the biological processes that our bodies carry that let you deliver these payloads. And each one is dependent on the disease and which cells in which the lights are burnt out that you're trying to go back in and correct.
So basically, gene therapy research is investigating a diverse group of tools that deliver genetic material with the goal of adding a working gene that was created in a laboratory to cells in the body. In short, it's seeing if we can use DNA to treat diseases.
The fields are growing and coalescing to the point where more and more diseases are having at least approaches developed to treat them more effectively than is possible at the moment. And where it's going to go in terms of non-viral systems and better viral systems and RNA-based systems, obviously a broadening technology, a broadening field. The old, interesting, we now even call the original approaches with find the disease, find the gene, put it into a virus, put the virus into people. That's sort of now jokingly called classical gene therapy. So it's become classical already, even at this early stage.
Gene therapy research has advanced much quicker than scientists had originally predicted. And as we've heard from these researchers about what it's like to be on the forefront of new ideas and scientific discovery, the patient was always on everyone's mind. Imagine what it would have been like to work on something for decades and finally be able to treat a patient. Dr. Samulski shares his story.
Connor was the first patient to get treated with our drug for Duchenne muscular dystrophy. Connor came down from Connecticut to get injected. And as usual with all of these things, you sit on pins and needles and you stay up all night worrying about if there's something we missed or something we should have done or another experiment, blah, 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 blah. And at some point, there's nothing else, no other rocks you can overturn to see if there's a risk that you missed out on. So we kept our fingers crossed, said our prayers, and Connor, who got injected with 10 billion trillion FedEx trucks delivering the gene that Xiao designed for dystrophin into his body, was playing with his video game. And his doctor said, hey, Jude, he's asking for another dill pickle. This is his third dill pickle. Is there something going on here? Let's leave him alone, Eddie, leave him alone. If that's what he wants, give him what he wants. But an hour after it was done, we were able to go in and visit with him, and he got through the night, and we were keeping our fingers crossed. And then all of a sudden, a week or 2 weeks later, it looked like he was thriving and doing well. And then we were getting reports from the parents like, oh, he's starting to go up steps. He looks like he's got more stamina. I mean, all of this was just earth-shattering to us in the sense that, you can only watch the rocket go so far and then it goes out of sight. And after that, you're relying on other means to determine if it's on the right trajectory to the moon and so forth. And so we were humbled and at the same time exhilarating feelings. And I think it was Connor, when he was going from 2nd to 3rd grade, don't quote me on that, he was asked to write what was the most important thing that he did or happened to him last summer. And he wrote that he was proud that he could run faster. Very impactful, and I frame it and I have it over my desk.
Next time on It's in the Genes, a Gene Therapy podcast, we'll dive into the research and how researchers are accounting for safety, the national organizations involved, and the investigational challenges that are creating hurdles for scientists. Stick around as we explore the wonders of gene therapy research. It's in the Genes, a Gene Therapy podcast is written and directed by Amy Board, produced and edited by Greg Holzman, artwork by Christina Newhart, post-production support from Josh Bragg, Rob Bradford, and Avra Friedman. It's in the Genes, a Gene Therapy podcast is produced by Bloodstream Media and presented by BioMarin. Visit hemedifferently.com for more educational information on gene therapy research. Subscribe, rate, and share It's in the Genes, a Gene Therapy podcast. Referrals from you are the best way we can reach new people. Thanks for listening. My name is Amy Board, and we'll be back next week with episode 2 of It's in the Genes, a Gene Therapy podcast.